And so this is what we often see, and beekeepers have often told me that there's an increased amounts of ball brood, and we now know this is a good sign. And this is active. There are bees that are investigating the colonies, and these often occur in clusters. We think this is due to the fact that the infested cell, it's very hard to detect these odors, and so they're uncapping cells in that particular area. Okay, well, welcome everybody, and good morning from Hawaii, and good afternoon in Michigan and where everybody else is at. So I've been studying, well, Varroa, I suppose, now for about 35 years, most of my life. I started with a national bee unit in the UK, then moved back to universities. My first job was to look for to help the beekeepers with monitoring the mites and finding treatment thresholds. And then with Brenda Ball, we discovered that it was the virus that played a big role in this, and that's what actually kills your colonies. I've done quite a lot of reproduction. And more recently, I've been looking into natural resistance, or what some people call it tolerance. But when I call colonies, when I'm talking about resistance, which is today's talk and how to select for them, I'm talking about bees that require no treatment whatsoever. So it's not reduced treatment. This is no treatment at all. So let's see.
Dr. Martin, we got your window screen. We can still see your whole window. Are you able to just share the slides, or it's showing your desktop?
It's not showing the slides.
Well, it is showing the slides, but it's showing your computer laptop screen there.
Ah, right. Okay. I'm all right on this side, but let me just check again.
Oh, maybe that's just the way that it's presenting, which is fine. It works. It's not a deal breaker.
Okay. Right. So that's on full screen for me. And yeah, you can see my the bottom line of icons, which I don't think I can.
Okay. Go ahead and proceed.
Okay. Right. So next slide. Okay. So what surprises most people is that Varroa resistant honeybees, Apis mellifera, have been around since the 1980s. I was studying them in the 1990s in Mexico. These were commonly known as Africanized bees, but they're still Apis mellifera, as we'll find out. And by the 2010s, in fact, they were very widespread. So people throughout all of sub-Saharan Africa weren't treating, throughout all of Latin America, up into Mexico, weren't treating, and their bees were doing fine. In fact, their honey production was increasing. They could sell organic honey. So this was, it was great for them, but it was in the Northern Hemisphere where people were treating. It wasn't really, resistance wasn't really thought about or something that we could do. And there was huge selection programs from the USDA and also in Europe. But more recently, in the last sort of 20 years, there's been several Varroa resistant, well-studied populations. Tom Seeley's in the Arnott Forest, and we've had them in Norway and Europe and France and Sweden, now in the UK, and you have several in the USA. And then also we've got this group out on Hawaii. So there's been an increase in the awareness around Varroa resistant populations. And so we did a big survey in the UK from where I'm actually based quite recently, because a lot of beekeepers there, well, not a lot, a minority of beekeepers were saying that they hadn't treated for 6 or sometimes over 10 years, and they were getting on with their beekeeping just fine. But they were very reluctant to speak because well, it still is the case. If they actually say they haven't treated, they can be classed as a bad beekeeper. They can be ridiculed by the rest of the people in the association as somebody who's going to create mite bombs. So these beekeepers have either not, they've left associations, or they just don't mention what they're talking about. So we did an anonymous survey to try and find out what was going on in the UK. I think it's got about 3,000 beekeepers involved, which is about 10% of the UK estimated number of beekeepers. And we found about 10% of them hadn't actually been treating for over 6 years. And it's a good guide. If you've been treatment free for Varroa for 6 years, then you almost certainly have resistant bees. And we have people that are up till, well, one beekeeper's over 20 years, but there's several that are well over 10 years now, and they're scattered all around the country. There's most associations in the UK have some people that haven't been treating for many years. There's one large area in North Wales where they noticed that the feral colonies were coming back after Varroa, and they switched from treating their colonies to collecting feral swarms and hiving them, and then not treating. They didn't know what was going to happen, but the colonies didn't die. They then, in their association, started to spread this information, and now we have about 100 beekeepers keeping about 500 colonies in this area in North Wales, and it's the largest single area where people are not treating at all. And we have new beekeepers in this area that are given nukes from existing beekeepers and told not to treat. And in fact, some of them actually don't even know how to treat the new beekeepers, which is a sort of a new dawn for this area, which is quite nice. So this is the biggest area we have in the UK. So I just want to jump quickly to a place that, unfortunately, that's caused me a lot of hassle getting back into America. But a couple of years ago, we went to Cuba. Now, Cuba is probably the best place I have ever been in my global travels for beekeeping. It's a totally amazing place. The forests there are full of palms. So they're palm forests, they're not classic trees. And the bees, absolutely, there's an unlimited supply of nectar and honey in these, nectar and pollen in Cuba. They have a centralized system. So they have about 220,000 colonies at the moment. It's braced up into associations in each area. They don't tend to move bees around very much. Each area has a queen in place, and they are selected for honey production and calmness. And they are extremely calm bees. They're some of the calmest bees we've ever dealt with. We don't usually use veils when we're there doing the beekeeping. And they got Varroa about over 20 years ago, and they decided at a sort of a central level, just like South Africa, that they wouldn't treat. It's not really an option sometimes because it's very difficult for them to import things. So they decided just not to treat. Because of the embargo that's on Cuba, they still have European honeybees, even though they're in the Caribbean. And most of the Caribbean islands have a mixture of Africanized or some European, but Cuba is just completely European. They have the Varroa mite that came in through one of their ports 20 odd years ago. It's a Korean type. So it's just the same as us. And surveys have shown that it's got deformed wing virus. And we know from some recent work that we've done that it's actually now, it was the A strain and it's changed to the B strain. So basically they have their situation is exactly the same as anywhere else where Europeans are kept. The big difference there's 20 years of no treatment for Varroa, and they have extremely good honey production, about 70 kilograms, which I think that's about 140 pounds per colony. And that's an average of them 220,000 colonies because they have data. And in good locations, they can get in excess of 200 pounds. So these colonies, despite being resistant, are actually extremely good gatherers of honey. So what I've been studying in the last sort of 5 or 6 years. And the reason we went to Cuba was to understand the mechanism. It's very clear that the beekeepers in this case have been ahead of the scientists. They've been keeping bees for decades now. We know part of the story, but until very recently, we didn't know the sort of whole story of Varroa resistance. And then understanding the mechanism, I'll go on to say how we can actually select for that. And so what we need to what's important in the story is, first of all, the virus, deformed wing virus, because as we'll see, without the virus, we wouldn't really have had a problem. Obviously, the Varroa mite, the honeybee, and I know that beekeepers are very attached to their honeybees, and they will tell me what color they are and what race. And we're very fortunate that this mechanism will work in all honeybees, whatever the race. And it works for all beekeepers as well. So this is, the backyard beekeepers, commercial beekeepers, because there's a big variety of beekeepers. And then the main mechanism is hygienic behavior. But the hygienic behavior is not the behavior that most people associate hygienic behavior with. It's sort of been hijacked by the freeze brood kill and pinprick tests and their reactions to removing dead brood. And of course, Varroa don't kill the brood, because if they did, they couldn't reproduce. They need to keep the developing bee alive so the mites can reproduce, and then the mites can emerge with the bees. If they kill it, then they don't reproduce, the vector dies and the whole cycle dies out. And that's why certain diseases like acute paralysis virus, we very rarely see. We see a more chronic virus, which is deformed wing virus, is the one that's taken over. So all these fit together. Now, we need to fit all these together. So the first thing I'm going to talk about is something that sort of comes up at the end, but I'm going to tackle it now, is grooming. So grooming is something which has been, in my eyes, has been anthropomorphized, as in we can see the mites on the bees, and so therefore the bees can see the mites, and why don't they move them off? And they should be able to groom them. And this has started a huge amount of work done by beekeepers and bee scientists, and some of it continues today. However, this originally started with Apis cerana. There were some original studies in Apis cerana, the natural host of Varroa, showing that grooming was very effective. But the original studies were flawed because they were using mites from Apis mellifera and not from Apis cerana. And as we now know, they didn't know then, but as we now know, that the chemistry, how they detect these mites, is extremely different. And when the work was redone with Apis cerana mites on Apis cerana colonies, the grooming was actually very inefficient. And in fact, one of my students went through all the literature and found that there's only 3 really good studies in Apis cerana, and the grooming is not significantly different in Apis cerana from the levels we find in resistant Apis mellifera or even in treated Apis mellifera. It's all very, very low. The things that are really stand out are mite infertility, as we've known for a long time. That's very high in Apis cerana in worker cells. It's also very high in Apis mellifera. We've known this since the 1990s, and it's not very high in treated colonies. And brood removal is a key thing. So the reason that grooming is not an important factor is based on the chemistry. But this is a really good example here, before we get to the chemistry, of something that we have seen as beekeepers and bee scientists and misinterpreted it. So if anybody looks carefully at Varroa mites, occasionally you'll see these dimples on the mite, and it's really clear on the right-hand side. And this looks, to all and purposes, like a bee has grabbed a hold of that mite and tried to sort of remove it with its mandibles. But an acrologist actually noticed that sometimes these were in parallel. So there was 2 dimples in the mite. And what he realized, that this is actually a birth defect. This has got nothing to do with bees at all. And it's where the muscles are that control the legs. And if there's not enough hemolymph in the mite, these when the muscles contract, they can cause these dimples. And what we did is they started looking at mites that were in cells that were reproducing, so the next generation, and some of them had dimples and they've never been exposed to them. So in fact, he was correct. So this is something where we've misinterpreted what we've seen. But the big problem with the grooming is that although we can see the mites, which is clear on the right-hand side here, the actual bees don't use vision, and inside a colony, it's pitch black. So they use chemistry. And when we actually looked at the chemical recognition or the chemistry of the mite and the bee, which is on the left-hand side, it's like a barcode effectively. The mites are brilliant at mimicking the chemistry of the bee. And to all intents and purposes, when a bee antenates a mite, all it sees is another part of the bee. It's a really common tactic used by parasites. The brawler fly, which was around before Varroa came, uses the same tactics. So even though these flies sit on the queen, and we can see them really easily, the actual bees couldn't, and that's why they wouldn't remove them, because they could easily do that. And this ability to mimic and to change their chemistry to fit the colony or to fit the particular bee that they're on, because foragers and nurses smell slightly different, is highly developed in these mites, and it can only take up to about 3 hours. And I had a PhD student study this for quite a long time. And what she came up with this small animation. So if you've got, say, a bee drifts in from another colony and it contains a mite, you can't really see the mite because chemically it's identical to the bee that it came in on, but it can be slightly chemically different from the bees that it's come into, or it could be a nurse that's emerged into a colony, which again is slightly different to the foragers. And when the mite moves to another bee, then chemically it's slightly different for a period of time. It's about 3 hours. However, it has, and at this point, it could be exposed to detection because it's smelling slightly different. However, it's got hooks and claws and the ability to go under the plates. And it only needs 3 hours to basically disappear it back into the colony. And then it's completely invisible to the bees. And this is why it's very, very difficult for the bees to actually see them because they're highly developed. And this is why grooming is not doesn't really play a role in the resistance behavior that are found in bees. And she called it the ghost in the hive simply because it was very hard to see, even though we can see them. So that aside, now I'll start talking about the mechanism. So first of all, we got deformed wing virus. So this is the really big player in this story. So when we first started to put all this together decades ago, there was a hypothetical situation where we would have just mites without the virus. This must occur at some point, and we think it occurs in the first few years when Varroa arrives. But now we're very fortunate. There are 3 cases. There's a small island off Brazil where basically there is no deformed wing virus. There are European bees that were taken onto this island, this Brazilian island. It's about 300 kilometers off Brazil in the Atlantic Ocean, called Fernando Noronha. It was going to be set up as a queen rearing station. That sort of failed, but it had European bees there, and it had Varroa, and they had constant mite levels for about 25 years. Well, it's longer now, about 30 years, and there was no losses. And when we went there and we checked, we were trying to interest to see why these colonies have survived. We found that actually deformed wing virus was absent and mite resistance had appeared. And this really is an opportunity for somewhere like Australia, where again, the surveys have shown that the virus is absent. And without that, bees can tolerate huge numbers of mites. The largest number of mites that I've ever seen or heard about is about 25,000 in a healthy colony. So without the virus, the bees can tolerate huge numbers, many tens of thousands of mites, and that buys them time to get this resistance. Once the virus appears, then that kills the colony quicker than the bees can develop the resistance, and you'll see how that comes to play. On the island, we had a look at their mite reproduction, And basically, it's extremely low. It's one of the worst reproducing mites that we've ever seen. And they're only producing about half a new female offspring per reproductive cycle, and they only reproduce 2 or 3 times in their life. Brood infestation could actually be quite high. There is a lot of mites, but most of these are not reproducing, and because they're not transmitting the virus, it has no effect on the colonies. They're extremely calm colonies. This is the Lydia and Emmy, my student from Brazil, used to working with Africanized bees, and this is the bee suits they wear on Fernando and Noronha. The bees are very, very, very quiet. And also there's 2 new cases now, one on the Solomon Islands and one up in Papua New Guinea, again, where they don't have deformed wing virus. And so the bees have developed resistant. Unfortunately, that's will never happen elsewhere in the world, because as soon as the virus appears, which is in red in this slide, the mites start a new transmission route, and that's what causes the problems, particularly when they feed on the pupae, because it shortens the life of the pupae, and that's what causes colonies to collapse, mainly over winter time. And the adults, they do become infected. Their life can be shortened slightly, but they mainly become carriers. So the virus now is present in pretty much all the colonies where Varroa are. It is transmitted by Varroa, but it's also now transmitted naturally by bees through their eggs, through the sperm. Fortunately, the levels that are transmitted naturally are low enough not to affect the colony, so it's still just the mites. And just like COVID, the virus has been changing, so it changes strains. It used to be the A strain, which was completely the only strain of virus in the Americas. But now it's changing over to B, which is more transmissible because it can replicate in the mite. In Europe, now it's pretty much all B. It's changing to B in Hawaii, and in America, it's going from A to B. And there are other strains, C and D now, which we don't really know much about. So that's all I'm going to say about the virus. It's there, and it's something that we need to consider. But what's really important to understand is the big difference between treated colonies or resistant colonies, or in this case, Africanized, and there's something to do with their reproduction. So this is a very, very simple example, but in susceptible colonies, 1 mite goes in, you get 2 mites coming out on average. And in resistant colonies, it's more stable. 1 goes in, 1 comes out. So you've got a stable population in resistant colonies. You have an expanding population in susceptible colonies that need treating. And we've known this for decades, ever since the sort of 1990s, but we didn't understand the mechanism of how this actually occurred. We knew infertility was important. And we did a very large study looking at a huge number, in fact, over 100 studies nearly. And we worked out in susceptible colonies, the mite infertility is about 17% on average. And in resistant colonies, it's very high. Now, this follows on from some work we did back in the 1990s, but it's, subsequent work has shown that. But we didn't know how this mite fertility came about. And this was the real missing sort of part of the story. And then there was 2 sort of parallel investigations. There was the Harbo and Harris work that came up with the very sensitive hygiene behavior. And then there was Melissa Rode over in Norway that basically discovered that when this sort of recapping behavior, which was effectively sort of the same sort of thing, but sort of independently found. And what we believe happens, and most of this is based on Marla Spivak's excellent work in hygienic behavior, is that the bees, we know there are a small group of bees which are highly sensitive. Let's call them sniffer bees between sort of age, I think it's age 10 and 15. They have very high levels of a brain transmitter, and they become sensitive to odors. And there's a big French team which have shown that we think it's the mite offspring produce a unique set of odors in which are ketones and acetates. And these sniffer bees then open up the cell. They make a small hole in the cell when they're trying to find the infested cells. They make errors, and so you have holes in neighboring cells, which tend to be smaller, but once they've opened up the cell, they leave the cells. So now you've got a comb with cells which can have holes in them, and we call these ball brood, or beekeepers call them ball brood, and they're left. Now, there are other bees in the colony that do two different jobs. One set of bees are called recapper bees, and they will come along, and if they find there's a hole, because it's accidentally, say, been damaged in some way, and the pupae is still alive and healthy, as far as they're concerned, they will recap it over. So they'll just basically reseal that cell. However, there are other bees that will come along, and if they find that that cell's infested, then they cannibalize that cell down. So basically, they will remove the infested pupae by cannibalization. They don't throw it out. During that process, the female mite, who's highly evolved and chemically mimics, actually escapes from the cell. So she can get onto one of the cannibalizing bees or just leaves the cell. So she's not killed, but she loses all of her offspring. So the offspring, any males or deuteronymphs or protonymphs that are developed in that cell basically get destroyed because they cannot live outside the cell. And so this causes the mite to reduce its lifetime reproductive ability. And so this explains quite nicely why there's high infertility, because if these mites are getting disturbed all the time, they're effectively becoming infertile. Mites can live almost a year, because that's how they're adapted. In Apis cerana, they can only reproduce in drone brood, and drone brood in some populations only produce once a year, and the mites need to be able to survive on the phoretic phase that entire year. So these mites can actually live quite a long time, but they can run out of eggs quite quickly. And so this is what we often see, and beekeepers have often told me that there's an increased amounts of ball brood, and we now know this is a good sign. And this is active. There are bees that are investigating the colonies, and these often occur in clusters. We think this is due to the fact that the infested cell, it's very hard to detect these odors, and so they're uncapping cells in that particular area. And when I'm talking about a recapped cell, this is what they actually look like. So on the top left, we have 3 cells, which are just normal sealed worker cells. Top right, if you open the cell up and look on the inside of the cell, it's all silvery. And this silvery is due to the reflection of the cocoon that's been spun by the larvae. And that cell's not being disturbed. The 2 lower cells, you can see that it's not silvery all across. There's a sort of matte, waxy, browny area in the middle, in the central area, small on the left-hand side and bigger on the right. And this is an indication that the cell at some point has been opened and then reclosed again. Unfortunately, it's very difficult to see this from the top, but it is actually quite easy to see from underneath. And this is a technique that's not really useful for beekeepers, but it's very useful for bee scientists, because we can use this as to work out the ability of the bees to detect infested cells. So when I talk about recapped cells, this is what I'm actually talking about. And it's really good evidence that bees can detect infested mites. So we've done lots of work on recapped cells, on worker cells. And if you look at places where there is no Varroa, so Varroa naive populations like Australia, well, we did this work before the mites turned up, a small island called Colonsay in Scotland and the Isle of Man in England, where they don't have Varroa, recapping is really rare. You can find it. They will recap cells in response to wax moth. So this is something that they already do. It's in their sort of arsenal of hygienic behavior. And if you have wax moth in a colony, you'll see that if you actually look, you could see that the cells are recapped, and usually in a fairly straight line as the wax moths move through. In Varroa susceptible populations, so those that have been treated, and this is work from Norway, Sweden, and 2 populations in France done by Melissa Rody, and then some work that we followed up in the UK. There are now 2 bars. So the red bar is indicating the infested cells which have been investigated. So the bees have opened them, but then they've been closed, and the non-infested cells which are in yellow. The key thing here is that in every population, the red bar, the detection of infested cells, is higher than the detection of non-infested cells, despite the number of mites actually being much lower, the number of infested cells being much lower. So all bees, all in every population, appear to be able to target these infested cells, which is good news. But when we look at resistant populations, they just get very, very good at it. So we've looked at, so there's the ones in Sweden, which is the famous James the 007 experiment. A couple of populations in France, one in Norway, the ones in England, Cuba, where we visited, and then Africanized and African bees, which are still part of Nostalapis mellifera, but they've been resistant for a very long time. So we've got Scutellata and Capiensis, the 2 different races that live down in South Africa. And you can see that some of these populations, on average, they're hitting 60, 70% of infested cells are being detected. There's also an increase in non-infested cells being detected, you can see here. And we were able to explain that nicely because Isabel Grindrod, real genius student, she was able to show that the recapping patterns in these colonies are spatial. So there's clusters, and they're always clustered around the infested cells. So you can have areas where there's no recapping whatsoever, they're just not interested. And we think this is due to the difficulty in detecting the odors. So they're checking lots of cells till they can actually find it. And this recapping behavior actually doesn't happen once, it happens multiple times. So cells are opened and then closed and then opened and closed. But the key thing here is that some of them get removed. And so we've got some nice data now showing that in resistant populations, between sort of 60, 50 to up to 80% of infested cells are actually being removed. So some of this is done in South Africa, we've got in Hawaii and in Cuba as well. There's a huge variation between the colonies, but the controls are much lower. So this ability to remove these infested cells, which is causing the infertility. So the framework of resistance is sort of summed up here. So we get resistant colonies have an increase in mite detection. That leads to an increase in recapping, which is what we use. It's the easiest way to indicate that it's a resistant colony for a scientist. That increases in increased brood removal. That reduces the mite reproduction, which increases mite infertility, which we know. It also decreases the number of mites in the colony and the virus burden. So the level of virus in colonies drops, and that increases your colony survival, and hence why resistant colonies actually can survive quite well. And what's really encouraging is we were able to compare data that from sort of in the 1990s in Brazil and Mexico, and the average infestation level at that point, these are in resistant colonies, was about 20%. Now it's dropped to about 4%. So we don't know if this is the mites are changing their behavior and avoiding worker cells. We definitely know that they're going into drone cells in large numbers. So it looks like that there might be a change in the mite behavior. I'll go on to the recapping of drone cells because this comes up as questions, and they don't do it. It's a real hodgepodge. Some colonies don't do anything. Some colonies do. Some colonies recap more cells that are non-infested. And overall, there's no targeting of infested cells. So it just doesn't happen in drone cells. But what does happen in drone cells, which seems to be important, is that when the first batches of drone cells emerge, then the mites, because they seem to be waiting, because they're not going into worker cells, flood into the drone cells. So you can see the infestation level of drone cells is very high, especially when you've got lower number When you've got lower number of drone cells, the infestation level gets very high. You have very high cases of multiple invasions, and their reproduction drops. So it's almost like a way of drone trapping as well. So this may be why they're not recapping drone cells or not removing drone cells. They're targeting just worker cells. So now I'm going to quickly move on to what we've been doing to help the beekeepers out. So now we sort of understand the mechanism. What we were interested in is how beekeepers can then propagate this. So we did a queen swap experiment. So we were basically asking the question, is this resistance, is it a learned behavior like we see in bumblebees that learn to play football and you can actually teach them to do things? Or is it a behavior carried in the queens, possibly the drones? Because that means if it's a learnt behavior, the only way of propagating it would be by splits, because you'd need the workers to continue that know how to do it. If it's in the queens, then the queen rearers can do it, which is really good. So what we did is we took colonies that had susceptible colonies, and we had colonies that were resistant, and we swapped their queens around. Now, we did this study both in the UK and England, and because of various constrictions, obviously you can't take mites into a place where there isn't mites. We had to sort of adjust them in the different countries, and it was also carried out during COVID. But basically, we took some naive queens, so these were queens which had never been exposed to Varroa, and we re-queened resistant colonies, and we did this in the UK, and we also did it in Hawaii. And then we also did queen swaps, where we actually took resistant queens out of resistant colonies and put them into colonies with naive workers, and vice versa. And then we had some controls as well. To make it easy to understand, the naive results are in red, and the resistant results are in green. So the idea was, if it was a learnt behavior, then the workers, you had a mixture of workers, because when the new queen was produced her own workers, they would be the opposite to what the workers that were there. So if it was, you had a non-resistant queen, she would produce non-resistant workers, but they would be in a colony with resistant workers, they could learn, and vice versa. So that we could then separate out the two things. And what we found, first of all, was a number of phoretic mites. So the mites on adults was, at the end of the experiment, was much higher in the naive queens that had started off with resistant workers than the resistant queens that had naive workers and subsequently had resistant workers. Another measure was the level of infested brood. Again, it was much higher in the colonies with the naive queens. And then we looked at the removal, and we only did this in Hawaii because of COVID. And basically, we were up at about 70% removal in the resistant queens, but the naive queens was down at sort of 40%. Still a good number, but we know that that can go up to about 30% in completely naive colonies. But it was much higher. Again, in the control cells, they removed slightly more. And again, in the recapping, we had a higher recapping in the resistant colonies than in the naive colonies. It did increase. There might be some drift in the US. We had very low recapping levels for whatever reason. It is a variable trait in the UK, but it was all in the right direction. Oops. So what that basically said was that if you take a queen from a resistant population and put it into any colony, then it will become, the workers will become resistant. We don't know the role of the drones in this. These are mated locally, so they were mated with resistant drones. But so somebody who has a resistant population can then propagate them by the queens. The mechanism, I think we might hear later on, but it's probably an epigenetic mechanism because of the way that it's been passed from workers onto the queens and become hardwired in the queen. So if you're going treatment-free, the people in the UK, there seems to be three main ways they're doing this. You just don't treat and let the bees sort itself out. It's probably not a good idea. People have lost colonies. People do get away with this occasionally. You can collect or buy resistant bees. I would not advocate buying resistant bees based on freeze brood kill, because you are selecting for a different behavior there. Or which some people have been doing, you can reduce your treatments over time, increase your monitoring. I'll then do some selection. And I think Randy Oliver's been doing this in the States, and over about 5 years, he's managed to get some resistant bees, resistant population, and it's happened in the UK as well. What we're suggesting in the UK is if people want to end Varroa treatment, then basically is to try and keep it simple, is to reduce your treatments that you're doing at the moment. Most beekeepers in the UK are about 75% are still actually treating, and they're not monitoring anymore. They just treat once or twice a year. And I think overtreatment's quite common now. But you need to half your treatment and reduce its effectiveness. But you also need to increase your monitoring, because if you don't increase your monitoring, you could, to avoid losses. You could conduct IPM, the wrong way around, integrated pest management, which is basically treat when you need to, if people are wanting to transition from one to the other. And lots of, everybody will have their own ways of doing this. One of the guys in the UK has been using a sort of selection program, and they've been quite successful. They've been using mite numbers on floors, but also this evidence, they're looking for evidence of pupae being removed, this removal behavior. And you can see this on the floor here, where you've got bits of legs and antennae. It's not all eaten, a lot of it's just thrown down, and you can detect this. And what they simply do is each year they monitor their mite numbers, look for these other characteristics, the ball brood, the cannibalization, and colonies which have got higher levels of mites, they don't breed from these, and they select from their lowest levels of mites. And after 5 years, they've managed to be able to get, most of their colonies have got stable mite populations. So for me, the do's and don'ts, I think the common with most people now is just use local bees. If you can get resistant bees from a local beekeeper, great. If you've got a big feral population, the feral populations have bounced back. This is how Oahu in Hawaii has basically become almost completely resistant. We lost tens of thousands of feral colonies. They came back after about 5 to 7 years. And now the beekeepers are just collecting the swarms from the forest and don't treat. And so most beekeepers on the island are not actually treating. And those that do, actually, their bees are resistant anyway, by the looks of it. Measure recapping, that's just, it's not an easy thing to do. But just start looking for these things. It's an easy thing to see the ball brood. And if you look carefully, you'll be able to see cannibalized pupae. And sometimes you can even see the mite fecal matter on the wall. It's not a good idea to move bees, especially resistant bees, into an area where people who are treating, because you can get mite reinvasion, and that can cause colonies to then die. Because I've seen situations where you have a large influx of mites into a single resistant colony. They are doing the behavior and removing the pupae, but if you get 40 to 60% of the pupae being removed, the colony just dies, because it's just not, it's killing too many of its own future workers, and it collapses. Yeah, don't guess. And my advice to people that are thinking of going down this road is basically not, is don't stop treating, because we don't know. It's hard to tell whether colonies are, where they sit on the level of resistance. The only way to do that is to actually monitor your mite numbers. For the beekeepers in the UK, they were wanting information. So we put a lot of information on this website called varroaresistant.uk. It's all free to use. There's a lot of scientific papers from people from all over the world that have been studying resistance. There's a lot of case studies by beekeepers. It's all UK focused, but it's the same traits, same ideas, what they do. I'm sure you can do it in Michigan. And some of the science we've converted into sort of beekeeper friendly literature, and there's now sort of several books. but most of the information is contained on the website. And then there's been a huge team. So I'm just the sort of spokesman for this. But we've had a whole bunch of people studying the viruses and the resistant bees. And all this work is largely funded by the British Beekeepers or Bee Disease Insurance, and we've been able to leverage funds. The work that I'm doing in Hawaii was funded by APSM. So, I think that's me done. I've got 15 minutes to try and answer any questions, and I'll stop sharing my screen.
Thanks, Dr. Martin. We got a little bit of a list of questions going, so if you'd like, I can go ahead and tee them up for you.
Okay.
This is from Anthony Antonucci. He says, in your research and travels, have you encountered the presence of tropolaelaps, and how your Varroa research findings can or will be applied to Apis mellifera in dealing with tropolaelaps, if you would dare to go down that one?
Okay. So yes, I have worked in Pakistan for a period of a few weeks, and there we did encounter tropolaelaps. Yes, tropolaelaps outcompetes Varroa mites, so it's a slightly bigger problem there. But I've been around bee research for very long time, 35 odd years now, and tropolaelaps was one of the sort of threats that was on the horizon 35 years ago, and it's still, people still worry about it, but it hasn't made that jump at all. The beekeeping that we have now is very different to what we had 35 years ago. And in fact, even, it's illustrated by even in Hawaii, where we've had mites here for sort of nearly 15 years. They haven't jumped to the other islands because people are a lot more conscious, and beekeepers are a lot more conscious about moving bees and queens willy-nilly around the world. Things do happen. But tropolaelaps, we still don't know its full biology. It can only reproduce in the brood. The bees, Apis dorsata, that it lives on migrate, so it doesn't go with them. There's no phoretic stage, and then it just reappears. So there's still huge chunks of its biology that we don't actually know. I'm sure the bees will eventually be able to adapt to it in the same way that they've done to mites, if given the right opportunities. Because once we start treating, we remove also the selective pressure. But I've relegated tropolaelaps in my mind to sort of something that's not very good at moving around the world, unlike Varroa. So I wouldn't worry about it too much. It seems to have very, very specific environmental conditions to it to survive. I mean, it got into Korea and never really got established. So I think in the last 30 years, it would have spread if it was really capable of doing it. There's something holding it. We don't know what.
One comment is the camouflage or the chameleon nature of the Varroa mite is fascinating.
Yeah, I mean, this is not uncommon. I studied chemical ecology for a long time. This is used by a huge number of parasites. This mite, as I think we'll hear from other people, is highly evolved. I mean, it only lives on one host, and that's a big risk for it. But its adaptations to live on honeybees is astounding, and the chemical recognition ability is even more like gobsmacking than we first could understand. Because when we finished the 3-year PhD studies, the last discovery we made, and we never followed up, is it appears it can't groom itself, the mite. So we don't know how it actually takes up these chemicals that are used in recognition, these alkenes, but it selectively takes them up. So it's like there's all these chemicals swimming around, and it actually absorbs the chemicals, only selectively the ones that are used in recognition, these alkene cuticular hydrocarbons that are covered in all bees. So it is amazing how they do this. We don't know how it does it, but it does it, and it does it really fast. So it will change from a worker, the smell of a worker, to a forager, which are very distinct in just a few hours. Yeah, they're amazing.
Reminds me of a movie, I think, from the 90s called Mimic. It's like it was a horror flick. It was pretty terrible too. These moth-like creatures that could mimic humans and just kind of zap around undetected. I'm glad that we don't have that pest right now, but let's see what 2025 brings. Well, we still got to get through 2024. Maybe they'll show up. We'll see. So on the DWV map that you posted in your slide presentation, there was a green circle. Somebody was asking, what did the green circle indicate?
Yeah, so the green circle is deformed wing virus strain C. It's very rare. We think it's lethal, so we think that when it pops up, it just kills a colony. So that's why it doesn't persist. We don't see it very often. And because it's so rare, we do detect it, but it's so rare we can't study it. So we really don't know much about it. The original Egyptian bee virus was found to be form D. So it's now been called D, but that's disappeared. We can't find it anywhere in the world. So this, just like COVID, and this is how viruses continually adapt, is that they have they change their strains to become more transmissible in the system that they're working in, and that's why it's gone from D till A. C is an offshoot, but if it's lethal, it just can't persist. So that's why it's very rare. But yeah, well spotted. So we know very little about it, but that's deformed wing virus C.
Okay. Tiffany Horelic is asking, is there a way we can measure, like effectively measure recapping?
No is the answer. You can do it. I mean, we've got videos. It's not that difficult. It takes me about an hour to do 100 cells, which is what we do, and it's fairly obvious from 100 cells if there's a high recapper or not. So if it's like down below 20% or if it's above 50%, which is what we would call a resistant colony. The other thing you need to do, because it's clustered, you need to take the frame of at least pink eye. The easiest way to do this is to actually look for the bull brood cells, because if you find colonies with these open cells in, these are much more common in resistant populations. And then alongside that, you'll probably find it's got high recapping. So there is instructional, there's videos out there to do that if you want to do it in association level. If you're young, you can do it without a magnifying glass. I need a microscope or a binocular microscope or just one of these lenses. You do need good lighting. So one of these cheap table sort of crochet things are really useful, where you've got a good light so you can see inside it. It's not difficult. You just need a small pin or something and just cut round the edges of it. It's a bit fiddly at the start, but you soon get used to it. So it can be done. We would like somebody at some point to invent something you could do it sort of very quickly with light or something you could actually, because it would be a really easy one. But at the moment, Randy Oliver's tried to use wax strips where you can actually just, with hair removing wax strips. He swears it works. We've tried it. Sometimes it does. It's a bit messy. Sometimes it works. Sometimes it doesn't. So if anybody can come up with a really good way of doing this, we'd be really pleased. But at the moment, it's more of a scientific technique.
I think that Randy has an abundance of the strips around because he likes to walk around with his shirt off. But that's just one theory.
Yeah.
So. Can you explain a bit more about the potential epigenetic transfer of Varroa resistance?
Right. Okay.
Yeah. How's that one?
Yeah. So the thing is that this isn't proven by a long way, but I talked to one of the top geneticists in the world and asked him the same question. And the question is that workers do the behavior. So we know that. And they don't reproduce. So how does that get passed on to the queen? Because she's the one that's actually carrying that trait. And we know that from the queen swap experiment. And he answered it the same way that I was thinking about along these lines. And this was somebody who really knew what they were talking about. But again, it has to be proven. But the theory is that we know that social insects, so that's honeybees, social ants and wasps, they are the top epigenetic modifiers. So epigenetics is gene expression rather than the sort of hardwired genes. So just because you've got a gene doesn't mean it's actually expressed, and it's the expression that's important. And this is really easy to prove in honeybees, because a queen and a worker, her worker, is genetically identical to each other. And obviously a queen and a worker are completely different. And the only reason that they're different is because the queen has been fed on royal actin, which is a protein, and that is an epigenetic modifier, which basically switches on a whole host of genes to make her bigger, to make her produce more eggs, and change a chemical outputs. So, and they've actually been able to take this royal actin and feed it and create Drosophila flies, which are like queens, giant Drosophila flies, to prove that it actually works. So epigenetics can happen very fast. So this is where the DNA is wrapped around structures like tennis balls. It's the easy way of thinking about it. And basically what we think the queen can possibly do is, during her lifetime, if she's threatened by a disease, she can then unwrap and rewrap her DNA around these balls. So there's a different suite of genes as are expressed. And by doing this, she's been able to, the colonies which are really good at detecting, that have these sniffer bees and these remover bees, are basically, they get set. So that's a selective advantage to her. So that combination of genes expressed, and we know it's polygenetic for some, well, that means many genes are involved. There's some work that's been done just recently in France, is that when she gets the right combination of these, she sort of sits on it and says, well, okay, I'll pass that on to my next generation because this combination of genes expressed is really good. So she sort of juggles things randomly to start with until she gets to the right one by this unwinding and rewinding of the DNA. It's not something that we as humans do very, very well, but social insects do it brilliantly. So we think this is this epigenetic because it can happen very fast. And we know that this is, Varroa resistance can appear in sort of, between 5 and 10 years by selection or just naturally. So that's only several generations of honeybees, but they're already pre-selected because they already have this, most of these behaviors. So they're just adapting it slightly. So it may just mean some small changes epigenetically, which then gets locked into the queen. And once it's locked in, it can then become hardwired over time because of its huge selective advantage. So I hope, I know there's somebody talking later on epigenetics, and please correct me if I'm wrong, but I think that's the best explanation I can give at the moment, not being an expert in that particular field.
Everything's weaving together nicely. I love it when a theme comes together. This is a good question from Jan Lawson. Would testing recapping behavior be redundant in VSH stocks that are being tested, let's say using the Harbo assay?
No. So we've actually had a look at some VHSBs, and they're not performing that well on the recapping thing. But this is a group that we've looked at. So from the work that we've done, so everywhere we've looked at, these are completely resistant bees. And I understand from some beekeepers in the United States that they have VHS lines, but they're having to then still treat. And that may be because they're in the environment of where other people are treating, or it could be that they're not, resistant enough. So when we've been looking at resistant, so these are truly resistant populations that have not needed treatment for a very, very long time. So this is, quite a long way down the line. So we're looking at the other extreme, that at a population level, not at a colony level, but when you look at sort of at least 3 colonies per apiary, then the recapping is always above sort of 40 to 50%, and it can be up to 60, 70, even 100% in some colonies. So it's a really good indicator of really high levels of ability to detect these cells. We then do removal studies, which are quite hard, actually, to be honest, and to do, and I wouldn't advise them, and usually find that we've not found any population that are not very good at removing and that have got high recappers. So I think it's another tool in your box. If you're using the Harbo assays, which I know you use widely in the United States, then you should have high recapping as well. And if you haven't, then I think you need to get there as well. But I think if you because it's a very tight, that the Harbo, the infestation level is very low that he's using, and most of the resistant populations we find, vary between sort of 4 and 10% infestation. They're actually naturally, resistant colonies are holding more mites because a lot of them are infertile than the Harbo assay is sort of advocating. So the selection pressure from the Harbo is very strong. So you should be getting that high increased level of recapping in them. So it's worth looking. If you think you've got good stocks, just have a look for it and it should be there.